Good morning, dear students. Today, first of all, I will discuss about the experiment where you have to study about plasmolysis and deplasmolysis. So, in this experiment, what you have to observe, you have to take one tradescentia leaf and you have to cut the inner portion of the leaf and then you have to put it under microscope. So, in that time, where what you will see that the cell will be in a turgid condition, it is water will be present over there, where you can see the lower surface. You have to peel with the help of a blade and then put it on the slide and put a drop of water, then see under microscope. What you will observe, a red colored content will be present inside the cell, that will be occupying the cell. Now you have to remove the water with the help of blotting paper and then you have to put some salt or sugar so that the plasmolysis will take place. The cell will be plasmolized, the protoplasm shrinks away from the cell wall. You can see the red colored content will be shrinking from the cell wall. Then remove the solution and after that add a few drops of water to the peel and examine after a few hours. What you will observe, the cell again regains its original position due to the deplasmolysis. So first we have seen the plasmolysis, where you have cut the lower portion of the leaf and then you have seen the protoplasm in the red color. Okay, afterwards what you will see, you will observe that when the plasmolized cell is again put it in water or some water is put over the leaf, cut portion of the leaf, after some hours you will see that the leaf again gets deplasmolized, means endosmosis has taken place. Okay, next we have to see absorption of water. So all of you are knowing that for the absorption of water, the xylem is responsible, and before the xylem, the root hairs which are embedded inside the soil, they are responsible. So the root hairs are the minute openings or the minute elongations from the epidermis, which is going inside the roots, inside the soil, the particles of the soil, and it is just absorbing the water from the soil. Okay. So you see, in case of roots, when you will observe, there is a zone of root hairs. Okay, afterwards we will see there is zone of elongation also. Okay, then zone of maturation also is there. So the first and foremost is zone of root hairs. So this is the zone which is responsible for the absorption of water. So you see, rate of absorption. So in case of root hairs, so first already I have told what is root hairs, and if we will see the thickness of the root hairs, so one root hair will be 0.1 to 10 millimeter long and 0.01 millimeter it is in diameter. So generally there will be 200 to 300 root hairs in a root. Okay, now if we will see the structure above the zone of root hairs, what you will observe, the rate of absorption just above the root hairs or the zone of root hairs, that will be minimum, because at that position there is suberization and lignification. So due to the lignification and suberization, what will happen, the root hairs just above the zone of root hair, there will be no absorption because there is suberization and lignification. There will be either no absorption or very less absorption. So what is lignification? Lignification is deposition of lignin. Lignin is a type of protein. So it will be present over there above the zone of root hair, and that is called lignification. It gives it a hard mass of solid. Okay, due to the presence of lignin, the cell wall will not be able to absorb any water from the soil. Okay. Now, suberization. Suberization is conversion of a cell wall into a corky tissue. Corky tissue already the cork cells that becomes very hard, sclerenchymatous tissue. Okay. Corky tissue by infiltration. Infiltration is something different substances, enzymes, hormones, it will enter inside it. That is known as infiltration along with suberin. So what will happen? The suberin will be infiltrated and the cell wall will be converted into a corky tissue. That's why it will not be able to absorb any water. So this is about the root hair. So the only zone of root hair is responsible for taking up the water from the soil and transferring it to the stem along with, so that it can carry to the leaves. Okay. Now coming on to the process, how it is happening. So you can see a figure is given in page number 45, figure 9. There you can see root hair cell, or simply it is given, what is given? The root is given. The different parts are also given. So the process is there. There is a simple process how the water will be absorbed. So first what will happen? The root hairs will be absorbing the water. Now next the cortical cells, cortex tissues, means the outside tissues of the root hairs that will be absorbing that water. Cortex cells. Then after the cortex, what will happen? The endodermis will be coming in action. So endodermis means the layer which is just inside the root hair layer. Epidermis, that is endodermis. So first is epidermis and afterward second layer is endodermis. Endodermis inside. So from the cortical cells, endodermis will absorb that water. Then we will have pericycle. Pericycle means that tissue which is connecting between the endodermis and the xylem. That is called pericycle. So in case of pericycle, the passage cells are also, it is also called passage cells. These passage cells will be responsible for carrying the water from endodermis to the xylem. So afterward, endodermis, then pericycle, and lastly the xylem vessels will be absorbing the water from the passage cells. So this is the order of passing of water. Okay. So first is root hair, then cortex, endodermis, then pericycle, xylem vessels, and simply by the root hairs will absorb the water by the help of osmosis, by the principle of osmosis. Okay. Sometimes by the active transport also the root hairs absorb the water in special cases, and mainly the root hairs absorb water by the means active transport takes place here for the absorption of minerals, okay, and nutrients. So minerals and nutrients, they are absorbed with the help of active transport, while the normal water is absorbed with the help of osmosis process. So this is an overall view of the absorption of water inside the roots. Along with one experiment we have seen, plasmolysis and deplasmolysis. Simple experiment was there. So I hope this part is clear to all of you. In the next class we will see the passive transport and the active transport along with ascent of sap. Till then, take care of yourself.